My name is Danni Li. For those who don't know me, I'm associate professor in the Department of Lab Medicine and Pathology in our department. So it's really my pleasure to have Dr. Ling Li to present her research. So I'm going to start with a brief introduction for Dr. Li. Dr. Li is a professor in the Department of Experimental and Clinical Pharmacology and the VWF Endowed Chair in Pharmacotherapy for the Elderly in the College of Pharmacy at the University of Minnesota. So she's local with us. So Dr. Li also serves as the associate department head and has joined faculty appointments in the neuroscience and molecular pharmacology graduate programs in medical school. She received her PhD degree in molecular and cellular and departmental biology from Iowa State University and conducted postdoctoral research on lipoprotein metabolism and atherosclerosis at the Rockefeller University and neurobiology of Alzheimer's disease at the University of Alabama at Birmingham. Before joining the university, Dr. Li was a tenured faculty member in the Division of Geriatric Medicine in the Department of Medicine at UAB Medical School. Her research has been focused on the connection between Alzheimer's disease and cardiovascular cerebrovascular disease, particularly on the roles of cholesterol and lipoprotein-related pathways in the pathogenesis of Alzheimer's disease and their therapeutic implications. Dr. Li's research has been supported by NIH and private foundations. She has served on several NIH study sessions, is a regular member of the Chronic Dysfunctional and the Integrative Neurodegeneration Study Session. As an educator, Dr. Li is also actively engaged in graduate and professional education, directing and teaching courses and mentoring PhD, PharmD, MD students, postdoctoral fellows, and visiting scientists, and serving on various committees. So today that she's going to tell us her research, one of her research direction, HDL, good for the heart, good for the mind, a lipid approach to stop the progression of Alzheimer's disease. Thank you, Ling, and please take it away.
Thank you so much, Danni, for your kind introduction. And good morning, everyone. And I really appreciate you joining us today. Beautiful morning. Finally the sun is out, and I think the temperature is going to be getting warmer this week. As Danni already introduced, indeed, our lab is focusing on the connections between Alzheimer's disease and cardiovascular and cerebrovascular disease, and in particular on the roles of cholesterol and the lipoprotein-related pathways in the pathogenesis of Alzheimer's disease, and also their therapeutic implications. So what I'm going to do today is that I'll start out by a brief introduction of Alzheimer's disease and highlight high-density lipoprotein, or HDL, and associated apolipoprotein, in particular ApoE1 and ApoE in heart disease and Alzheimer's disease. So after that, I will share with you two lines of research in my laboratory. One is focusing on harnessing the benefits of HDL and the mimetic peptides to prevent and treat Alzheimer's disease. And then another line is focusing on the role of a process called protein prenylation. So this is a lipid modification process in the pathogenesis of Alzheimer's disease and its potential as therapeutic target for Alzheimer's disease. Okay, so as all of you already know for sure, is that the Alzheimer's disease is named after a German doctor, Dr. Alois Alzheimer. He described the first case of Alzheimer's disease over 100 years ago in 1906, and the first patient name is Auguste D. So this slide shows a cross-section of brains, one from a normal individual and another one from the Alzheimer's patient. And I'm pretty sure you can see clearly that the Alzheimer's brain has lost a lot of brain tissue. So under the microscope, just like Dr. Alzheimer's described over 100 years ago, the pathological hallmarks of Alzheimer's disease include the amyloid plaques, which is composed mostly of the amyloid protein, beta amyloid protein, and the tangles, neurofibril tangles, that is mostly composed of the hyperphosphorylated tau protein. And you can see here, these are silver stained brain sections, and these are amyloid plaques, and these are magnified tangles inside the neuron. And notice this is actually the section from the first patient, Augusta D. So even though we have known the Alzheimer's pathology really for a very long time, but the pathogenesis of Alzheimer's disease is still not fully understood. And as a matter of fact, the biggest risk factor, unfortunately, is aging. As you can see here, for people who are older than 85 years old, the risk of Alzheimer's disease can be like over 50%. And because of the increase of aged people in our population, and the prevalence of Alzheimer's disease has increased very steeply. And as you can see, at current, we have over 6 million people in the US living with Alzheimer's disease. And it's predicted by 2050, the number will be doubled if we don't have any effective prevention or treatment for Alzheimer's disease. And unfortunately, so far, we don't. The good news is that in the past 30 years or so, there have been lots of genetic discoveries, many gene mutations, and also the risk genes are associated with risks for Alzheimer's disease. And as you can see here, the mutations in these three genes called the amyloid precursor protein, this is the protein give rise to that beta amyloid accumulated in the Alzheimer's brain. And also other two genes called presenilin-1 and presenilin-2, or PS1, PS2. So those are involved in the processing of the APP. The mutation in these genes are cause Alzheimer's disease, means if people have these mutations, they're definitely going to develop Alzheimer's disease. And the type but these are very rare. So it's causing what's called the familial early onset Alzheimer's disease. However, over 95% of Alzheimer's disease actually are sporadic. So that means that they are sporadic with not definitely a mutation or anything like that. However, in the early 90s, there were discoveries, very important discoveries made, was that a specific ApoE gene, is called the ApoE4, or the allele, is associated with a great increased risk of Alzheimer's disease, especially in the sporadic cases. So as you can see here, the homozygous situation with ApoE really is the highest, strongest genetic risk factor for sporadic late onset Alzheimer's disease. Even a single allele still increases very dramatically. So in humans, we have three alleles of ApoE. So ApoE2, ApoE3, and ApoE4. So this is caused by actually a single nucleotide difference in the gene, and then leads to a single amino acid difference between these forms. So in our population, the most prevalent one or most common one is the E3 allele, which has a low risk for Alzheimer's disease. And E4 allele is about 15% of allele frequency, but that is the one is the increased risk for Alzheimer's disease. On the other hand, the E2 allele is protective. So it protects people from developing Alzheimer's disease. And also, as you can see here, not only the E4 allele increases risk, but also have earlier onset of the age of onset of the disease if people carry the ApoE4 allele. So why the ApoE4 has a higher risk of Alzheimer's disease is not completely understood, but I want to point out is that ApoE4 ApoE itself is an important lipid cholesterol transporter. And the ApoE4 allele itself is associated with the increased risk of hypercholesterolemia and also atherosclerosis. And because it's a lipid and cholesterol transporter, so it is believed, and actually compelling evidence indicated that it must have associated with the lipid associated pathways. So indeed, over the years, it has been found that the ApoE4 isoforms or the level of the ApoE and also their lipidation status, because it's a lipid cholesterol transporter. And all of that could affect the A beta aggregation and also A beta amyloid beta protein clearance, and with other functions, which leads to the Alzheimer's pathogenesis. And more recently, Dave Holtzman's group also showed that not only ApoE isoforms affects the A beta, but also affects the tau tangle formation and also neurodegeneration. So even though there were really much of research going on with the roles of ApoE in Alzheimer's disease, and not much actually done with this cousin called ApoA1 in Alzheimer's disease. Both ApoA1 and ApoE, they bind lipids and form HDL particles or part of the HDL. So as you can see, along with many other molecules and associated enzymes, so the HDL particles, it circulates in the circulation, the blood circulation, and also in the CSF in the brain. But important thing is that in the systemic circulation, the main component of the HDL is ApoA1. So that determines the HDL function, and then that is actually protects against atherosclerosis and heart disease. But in the brain, the main apolipoprotein associated with the HDL-like particles is ApoE. Because of the HDL has so many different molecules, it also has multiple functions, including the ones we well known is cholesterol efflux or lipid efflux, anti-inflammatory, and anti-oxidative properties. So we all know it's very well established is that the high HDL cholesterol levels is protective against cardiovascular disease, and high LDL on the other hand is bad, is increased risk of cardiovascular disease. So here I'm showing the data from a famous study called the Framingham Heart Study, and basically it shows that if someone who has high LDL cholesterol, but if they also have high HDL, then their risk of cardiovascular disease can be still pretty low. But if someone has low LDL cholesterol, but has a low HDL, then their risk for cardiovascular disease could still be high, pretty high. Of course, the worst scenario is that someone has high LDL cholesterol, but also low HDL cholesterol. So you are, I'm sure, very familiar with this often called the good cholesterol, bad cholesterol, because HDL protects against cardiovascular disease. So it's being called the good cholesterol. And then the LDL, because it increases the cardiovascular risk, it's been called the bad cholesterol. So one of the reasons of the bad being LDL cholesterol is because it causes atherosclerosis, which is the buildup of the cholesterol plaques in the blood vessels, especially in the arteries, and particularly in the coronary arteries. And that, when it's completely blocked, it leads to the heart attack. So in Alzheimer's disease, besides the well-known amyloid plaques and tangles in the parenchyma of the brain, also amyloid actually accumulates and deposits in the blood vessels. And this pathology is also very prevalent in Alzheimer's disease. Up to 70–90% of Alzheimer's brains, you can find this pathology called cerebral amyloid angiopathy, or CAA. So it's a vasculature pathology. Not only happens in Alzheimer's, like naturally in the pathology, but also now it becomes an important aspect, because when we do the anti-amyloid immunotherapy, CAA becomes a very severe side effect for that treatment. So years ago, we've been asking that question: if the HDL is cardiovascular protective, can we harness this benefit of ApoA1 HDL and to mitigate memory deficits and amylopathology, and including the vascular changes in Alzheimer's disease. So what we did is we generated a triple transgenic mice across the APP PS1, which is the amyloid, model of Alzheimer's disease, and crossed with the human ApoA1 transgenic mice and produced 4 different genotypes mice. So we studied their behavior and also their pathology. But first of all, the Alzheimer's, the APP-PS1 mice, they develop just like humans, age-dependent of beta amyloidosis in the brain. As you can see here, 3 months, no amyloid plaques, but then started at 6 months, they started having some plaques. This is by immunohistochemistry against a beta peptide protein. And then as the mice age, the number of plaques increase progressively. And then as expected, the human ApoA1 transgenic mice have higher HDL, higher cholesterol levels. And as you can see, mostly in the HDL fraction. So with these mice, we first look at the behavior function, because as you know, one of the early signs for Alzheimer's disease is memory loss, and especially spatial memory loss. And Alzheimer's patients walk out the house, they really have difficulty find the way back. So neuroscientists used one of the tests called the Morris-Watermaze test. This is how it's set up. Basically, is assessing the ability of the mice finding the hidden platform in the water maze. So here are the results from this experiment. And as you can see, the 3 months, this is when they don't develop a pathology, everybody actually can find the hidden platform. But then by the 10 months, and you can see the Alzheimer's mice without the high HDL, they develop a deficit in finding the hidden platform. But then the ones with the ApoA1 expression still continues to form similarly as the non-transgenic mice, like the non-transgenic means the white type mice. And not only that, the learning process, but also memory. So this is when we take away the platform and then see whether they remember that location. So if they spend more time in that quadrant or cross that area, that means they have better memory retention ability. And as you can see, the overexpression of ApoA1 or increased HDL can rescue some of the deficit. And then, of course, we study Alzheimer's disease. One of the pathologies is amyloid deposition. So interestingly, when we look at the total overall amyloid deposition, there was no difference. But then when we use the Congress stain in trying to separate the parenchyma amyloidosis from the vasculature amyloidosis, which is CAA. And then now you can see the parenchyma amyloid, no difference, just like the overall, because these are very small portion of the brain. And then so now if you separate, and CAA, as a matter of fact, has a significant, almost half level in the ApoA1 overexpressing mice. So clearly that has significantly reduced the cerebrovascular amyloidosis in these mice. And because of the ApoA1 HDL has anti-inflammatory functions, so we also look at whether they have any effect on the neuroinflammation, which we use markers for microglia, which is called the EBA1, and then also for astrocytes and GFAP. And then their levels, it represents the activation status of these two immune cells in the brain. So as you can see here, clearly the GFAP level was significantly decreased, and we saw a trend in EBA1, but not significant. So we were puzzled in the beginning, but it turned out to be it's a dose phenomenon. Because ApoA1 has limited penetration across the blood-brain barrier, so you can see in the CSF, the level is quite low. And when we use a cell model, microglial cells called BV2 cells, and they have more ApoA1 in the system, and now you can see significantly decrease of A-beta-induced activation of microglia. So from here, we clearly can see ApoA1 HDL could be beneficial for Alzheimer's disease. However, ApoA1 have limited penetration to the, to cross the blood-brain barrier. So certainly for therapeutics, that's not practical. It's a big protein. So now that's why we move on to working with the small molecules, HDL mimetic peptides for to look at whether they have therapeutic potential for Alzheimer's disease. So they are series of ApoA1 or APO or HDL mimetic peptides. One of the most well characterized is called 4F. And you probably wonder why it's called 4F, because this is actually an 18 amino acid peptide. It doesn't have any homology, sequence homology with any natural protein, but it has this secondary amphipathic helices similar to apolipoproteins like these in ApoA1 and also in ApoE. So back to that question about 4F, is that because this molecule contains 4 phenylalanine amino acids in this, as you can see here in the hydrophobic phase. So the polar and hydrophobic phase, that's the apolipoprotein, they can interact with lipids and also interact with the aqueous environment. And we have the colleagues of my former colleagues at UAB develop a series of those peptides. Some of them have, like this one, have 4 phenylalanines, others have 2, 3, 4, 5, and 6. So 4F is mostly being characterized and showing similar or mimicking the functions of HDL, increased quality of HDL, and actually reduces atherosclerosis in mouse models, and actually already advanced to human clinical trials for cardiovascular disease. So that's why we choose 4F for our studies related to Alzheimer's disease. So because 4F is like HDL and apolipoprotein, it has been shown in the peripheral cells, it has cholesterol and lipid efflux ability, promoting formation of HDL. So the first thing we did is work with the primary astrocytes. So astrocytes in the brain are the main producer of ApoE and then forming the HDL particles. So what we did is that the ApoE needs to be secreted, lipidated to become HDL particles and to be functional. So what we found is that in astrocytes, a 4F peptide significantly increased the secretion and lipidation of the ApoE. So as you can see here, compared to what's called the scramble 4F peptide. So scramble is the control, means we have the same amino acid composition, but they are not in the order to, in the same order forming the amphipathic. So helices, so they don't have that structure required for apolipoprotein function. So these results has very significant implications because already mentioned, ApoE need to be lipidated to be functional. That's one aspect. Another important aspect is that ApoE4, which is the risk factor for Alzheimer's disease, have deficits in secretion and lipidation. Also want to mention here, we use the native gel, the non-denaturing gradient gel, to separate the particles which are based on their size, whether they have poorly lipidated, smaller size, or bigger ones which have lipidated. So you can see the difference here, but they don't form like very distinct bands as the SDS gels. Okay, so when we see that, the first question we want to ask whether this is specific for ApoE. So we also checked some other commonly secreted proteins by astrocytes, like ApoJ and also the complement C3 protein. And as you can see here, clearly it's quite specific for ApoE, and then because ApoJ and the C3 no significant difference there. So not only that, we continue the study, find out that the amyloid beta protein, which is the one precipitate in the deposit in the Alzheimer's brain, that protein has a dose-dependent effect on ApoE secretion and lipidation. It inhibits inhibits it from secreted and from lipidated. And then when we treat it with the 4F peptide, the HDL mimetic peptide, we can partially rescue that deficit. Now you can see more ApoE secreted, and also, in the secreted protein, they are pretty in the lipidated form, in the highly lipidated form. So then you would ask, well, you're using mouse primary astrocytes. What about human? Does it work in human astrocytes? Indeed, we also tested that in human primary astrocytes. And as you can see here, it is exactly doing the same thing, increased secretion and increased lipidation. And because we know in the periphery, ABCA1 is an important lipid and cholesterol transporter sitting at the, it's an integral membrane protein, intramembrane protein. And so that mediates the lipid and cholesterol efflux through the apolipoproteins. So we tested whether that's also required. So we used the CRISPR method to knock out the ABCA1 in the astrocytes. And as you can see here, clearly in the presence of ABCA1, and then it has a significant increase of ApoE secretion. But now after we knock out ABCA1, now the effect of 4F has been blocked here. So it indicates the required of ABCA1 in this process. So because of time, I don't have time to show you all the data here, but this is simply summarize what we found in this study. Basically, this HDL mimetic peptide 4F promotes the secretion and lipidation of ApoE, and the counteracts of the A-beta-induced inhibition of ApoE secretion and the lipidation in the primary astrocyte, both in human and mouse and in human. And this effect is through the transport, the secretory transport from ER to Golgi, which I didn't have time to show you the data, but this process, entire process requires the presence of the ABCA1 transporter. So because I mentioned about the ApoE 2, 3, and 4, this is really our goal, is to really to see whether 4F has any benefits when, in terms of rescuing ApoE4 deficit. So again, because of time, I'm not going to show you all the data, but just want to show you a very important piece of unpublished data here. So as I mentioned previously, ApoE4 has lipidation deficit, actually also secretion deficit, but I'm showing you the lipidation here. And again, as you remember the poorly lipidated, the smaller molecule here, if you look at E2, clearly most of them are in the large lipidated particles, very little or poor lipidated. E3 is somewhere in the middle, it's a little bit more, but then when you look at the E4, lots of them are poorly lipidated, which is exactly like what's in the literature has been shown, consistent with what's in the literature is that E4 has deficit in lipidation. And now this is in absence of 4F treatment. But now look at when we add 4F into the system. Now you can see 4F actually has restored the making both for the ApoE4 and ApoE3, or everybody now similar to ApoE2. So again, we remember these are native gels, so they look like a smear, but it's because they are all different sizes of lipidations, and so that's what we believe is a time probably dependent process. All right, so again, we want to be translational. So we want our results to be more translational. So we actually also now testing that in human iPSC-derived brain organoids. And this is in collaboration with Dr. Tim Breyer's lab in the Stem Cell Institute and others. They have developed a robust system to produce brain organoids from the iPSC cells. So we have different iPSC, different ApoE isoform specific iPSCs from collaborators from California. And then we have done some preliminary studies and showing that indeed in this organoids, there's an age-dependent increase of ApoE production, as you can see here. And we have shown that 4F also can promote the ApoE4 secretion in this system. So this is now we're going more studies in those and then looking at Alzheimer's related pathologies, and a graduate student is working on that. So at the same time, because we're working on trying to sporadic Alzheimer's disease, mainly deficit is in A beta clearance across the, one of that is across the blood-brain barrier. So in collaboration with Dr. Candy Mala's group in the pharmaceutics department, we actually use a cellular model of BBB to test what effect a 4F treatment could have on the A beta crossing the BBB. So you can see here, these are directions from L to A, means luminal to upluminal side. This means it's from plasma to the brain. This is like intake. And then the efflux is from upluminal to luminal side. And this is representing the efflux from the brain to the plasma. So as you can see, not a significant effect on intake, but you can clearly see for both A beta 40 and 42, which are the main species of A beta in the brain, and there are significant increase of efflux. So that it predicts that could help the efflux of A beta, removal of the A beta from the brain into the plasma for disposal. And at the same time, we also tested the in vivo, comparing the blood-brain barrier penetration of ApoE1 versus the small peptide. As I mentioned before, ApoE1 has very limited permeability across BBB. But as you can see here, 4F peptide has over 500-fold increase of penetration. So we would predict that this peptide will be more potent, more effective than the native ApoE1 in terms of alleviating or decreasing, reducing the amyloid pathologies in mice. So we are ongoing animal experiments to test whether this is the case. So I guess to summarize this part of the work, I would say simply whatever is good for the heart is also good for the brain. And as I showed you, HDL is good, but we believe HDL mimetic peptides could make it better. So I really, this is the connection of the heart and the brain for this part. And I wanted to switch gears now to share with you another main lines of research in my laboratory is focusing on the role of protein prenylation in Alzheimer's disease and its potential as a therapeutic target for Alzheimer's disease. So protein prenylation, as I mentioned already, it is a post-translational lipid modification process, and it's characterized by enzymes including the transferases, including the farnesyl transferase and also general transferase. So those are working on the protein which has a C-terminal motif called the CAAX or CAX box. So in this case, they use, as you can see, the fatty acids-like molecules called farnesyl pyrophosphate and the general pyrophosphate as a donor, and then lipidated the protein. So then they become farnesylated or generate protein. So they become more hydrophobic. So then they can associate with membrane, which I will show you a little bit later on. So again, you probably ask, oh, how come you're interested in this? This is actually go back to way back when we are starting work with a cholesterol-lowering drug called statins. And then I'm sure you're all very familiar with this. Millions people using statin to manage cholesterol levels. So statins turn out to be that it not only reduces the cholesterol synthesis, biosynthesis, the final product. There are so many intermediates in between because the statins acts on the HMG-CoA reductase is quite early in the pathway. So the intermediates, especially the ones called isoprenoids, and the main ones are the short chain, C15 and C14 chain. The isoprenoids called farnesyl pyrophosphate or converts to the general phosphate and FPP or GGPP for short. And so again, these were serving as the donors for protein prenylation. As you can see here, the FT or financial transferase or GGT, the general transferase, and they prenulated protein which has that CAC spot and turned out to be almost all the small GTPases need to be prenulated to be in the right place in the cell and then to function. So these are molecular switches, and they really play important roles in many different processes, even though it's not so much studied in cardiovascular and Alzheimer's disease or neurological disease. It's heavily studied in the cancer field because small GTPases like RAS and all these are involved in cancer development. So in all these processes, because they are not cholesterol, they're independent of cholesterol. So we believe many, and other studies also show many of those pleiotropic effects of statins, which are not related to cholesterol reduction, are actually mediated through isoprenoids. So as I mentioned, because this prenylation is so important to make the protein trafficking correctly inside the cell and associated with the membrane, and as you can see this cartoon here, just using RAS protein as example, how that's get prenulated, and together with the palmitoylation or other fatty acid the modification, and that gets them to the membrane localization, interact with their downstream effector protein, and then transduce those signaling pathway. So they are extremely important, and they're so important, actually, if you knock them out completely, the mice will not survive. Developmentally, it's extremely important. So we decided to study directly the role of protein prenylation in Alzheimer's disease. Because of the importance of, if you knock that out completely, no viable mice will be produced. So we decided to start out with the heterozygous knockout of the FT and GGT gene. So in this case, again, we use the APPS1 models of Alzheimer's disease. And when we look at those mice, when we cross the mice and then made the model, and we can clearly see that the amyloid deposition is decreased in both of the cases. Either you reduce the FT or GGT, but clearly appear to be reducing FT, you have more effective reduction here. And then we also look at the neuroinflammation. So again, with the GFAP and EBA1, look the markers, the astrocytes and the microglia, and you also see that both of them have attenuation of neuroinflammation. But again, the FT haplodeficiency has a more effective reduction of neuroinflammation. So then when we look at the cognitive functional effects, and again, use Morris-Waterman's test, in the learning process, you already can see the Alzheimer's mice has learning deficit, but then the reduction of GGT and the FT appear to be helpful somewhat. But when we look at the prototype, which is the memory retention, it clearly shows that the financial transfer FT clearly rescued the memory deficit, although there is a little trend with the GGT reduction, but it is not significant. So we find that there's a discordance between the pathology and the cognitive function. And it turned out to be that the GGT reduction has negative effect on the synaptic plasticity when we use the electrophysiology approach to look at the long-term potentiation of the neurons in the hippocampus. As you can see here, heterozygous deletion of the FT has no significant effects, but then in the GGT heterozygous mice, and then significantly suppressed of the LTP. And in addition, when we look at the dendritic spine density of the neurons, and we also found that the GGT heterodeficiency reduced those synaptic, those spines very significantly. So, and you can expect in the behavior test then the GGT heterozygous also have deficit in the learning deficit in these mice. So for the therapeutic, and you can see now we need to be focused on the FT pathway because of that doesn't have any negative adverse effect, but GGT reduction could lead to negative effect. And we believe that neutralize the limited benefits from the pathology reduction. So then we recently start study the human brain samples directly, to see the dynamics, the change of the granular proteins or related signaling pathway in the human brain at the different stages of Alzheimer's disease. So we obtained the postmortem brain tissues from individuals with either normal cognitive function or mild cognitive function or MCI or Alzheimer's disease. So we started out by looking at the levels, the protein expression of those enzymes, the farnesyltransferase or general transferase. So as you can see here, the farnesyltransferase is significantly increased. As you can see, a little bit increased in FT, but the trend increased, but clearly significantly increased in Alzheimer's brains. But GGT somewhat increased, but not significantly. So the next thing we did is look at the protein prenylation status. And what we choose, because it's an FT increase, we choose a protein is called HRAS, because this is a target of FT. Not only a target, it's exclusively farnesylated protein, because sometimes if you inhibit farnesylation, some proteins actually can go prenulated by GGT, but HRAS can only be prenulated by FT. So we use ultracentrifugation to do the subcellular fractionation and to separate the membrane fraction and the cytosolic fraction, because if the HRAS is farnesylated, then they would be located in the membrane. And then on the other hand, the unprenulated would be in the cytosol. So as you can see here, we not only see a significant increase of farnesylated or membrane-associated HRAS in AD brain, but also in MCI, so indicating that could be an early event in the pathogenic process. And importantly, the total levels of HRAS actually has no difference between different status of Alzheimer's disease. And as expected, the relationship between membrane-associated HRAS is positively correlated with the levels of farnesyltransferase, the enzyme. And then we look whether increase of the membrane association leads to any downstream effector protein or signaling activation of HRAS. And so one of the well-established downstream pathway is ERK pathway under HRAS. So the phosphorated ERK means the activated form of ERK. And as you can clearly see that consistent with the increase of the membrane-associated HRAS, and you see the activation of ERK pathway, not only in AD and also in MCI. And then when we look at the correlation, and then clearly the membrane-associated HRAS is positively correlated with the phospho-ERK to total ERK ratio. So from here, we postulated that upregulation of protein farnesylation maybe accelerates the process of Alzheimer's pathogenesis and in human. So is this the case? So you again go back to mouse model to test this hypothesis. Now remember that a complete knockout of FT or GGT is actually embryonically lethal. So this time we actually took an approach using the Quilox-P system to generate conditional knockout, especially in the forebrain neuron specific FT knockout mice, and then cross with the APP PS1 mouse model. So we use the 3 steps of mouse breeding, lots of mouse breeding, and generate many different lines, but we use those 4 lines for experiment. First of all, we need to confirm indeed in these mice, we can have what's called the region-specific and also age-specific patterns of the deletion, because we don't want to delete any FT during development, and this chemogulin kinase promoter only active after birth. So indeed, if you look at here, we use two markers now for phenylation. The well-established one is HDJ2, and then you already saw the HRAS is another phenylated protein. And then when you have inhibition of this enzyme, then you will see more unprenulated proteins. So indeed, they have different mobility on the gel. So you can see this bigger molecules here are unprenulated. And as you can see, these are only seen in cortex and hippocampus, frontal cortex, but not in cerebellum. And also, as you can see, when mice are getting older, this unprenulated protein get increased. So indeed, it's an age and region specific deletion of FT situation in these mice. So then we went ahead and looked at the pathology in these mice, and then this is the amyloid pathology, and by both immunohistochemistry and ELISA, and then we still show that they indeed have a decrease of the amyloid deposition accumulation in the brain. And we also use immunoblot analysis looking at the products of APP processing and found that this is partially caused by reduction of the APP processing and also the production of the A-beta peptide. So we also used unbiased transcriptomic RNA-seq approach to see at the global levels what changes in this brain after we knock out the neuronal specific FT. So you can see the APPPS1 mice, the mouse model has lots of genes upregulated. But then when you knock out the neuronal specific FT, a lot of them get downregulated. What we found is there are two top of pathways got downregulated by neuronal FT knockout, the cholesterol homeostasis cluster genes, and also in the mTOR C1 signaling pathways. So then we said this must be, the mechanism underlying what we see the benefits of FT in the mouse model. So we are actually using a cellular model to test that relation or validate that relationship. So we use the cellular model is the neuronal cell lines by white cells, but they overexpress APP. So as you can see here, when you have overexpressing APP leads to the upregulation of the downstreams of the mTOR pathways, and then those pathways, including like the ERK and the ERK pathway, and also the ribosome S6 pathways that significantly increased in the presence of overexpression of APP. So then we use a pharmacological FT inhibitor to see what happens if we inhibit this pathway, the signaling pathway. So first we verify that if we use a farnesyltransferase inhibitor, the one is called the tipifarnib, and indeed you can see clearly the increase of the unprenulated protein in these cell lines. And more importantly, now you can see with the treatment of the FT inhibitor, now you can see all of those pathways which were upregulated by APP overexpression is now have been reversed, either to the no change, no different from the cells which are not expressed, or to reverse to reduction. So we clearly validated that this is indeed through this pathway. So to summarize, again, I didn't show you all the data, but to summarize this part of the work is what we have shown is that upregulation of protein prenylation or FT, the enzyme, leads to the increase of the phenylation of those target proteins, including RAS or RHEB, another phenylated protein, and increase their association with the membrane, and causes hyperactivation of downstream pathways such as the mTOR C1, and these hyperactivation leads to the pathology of Alzheimer's disease, and then in turn this pathology further activates of the mTOR signaling pathway, and then forming this vicious cycle. But if we use genetic deletion or pharmacological inhibition, we can reduce the phosphatidylation of these proteins and basically break this vicious cycle. So then it becomes beneficial for Alzheimer's disease. So in conclusion, I hope I was able to convince you today that Alzheimer's disease and cardiovascular disease are closely connected, and dysregulation of lipid and lipoprotein-related pathways plays an important role in the pathogenesis of Alzheimer's disease as well as in cardiovascular disease. And targeting these lipid lipoprotein pathway could be an effective approach to defeat Alzheimer's disease. So I like to end my talk by quote this quote from the first Chinese medical text. So it says that superior doctors prevent the disease, a mediocre doctor treat the impending disease, and an inferior doctor treat the full-blown disease. I'm sure all of us would like to be the superior doctors. So hopefully we will be able to prevent Alzheimer's disease like we already can, before, like for cardiovascular disease. So lastly, certainly not the least, I have been really fortunate to work with a very talented group of people in my lab. And this is, all the good times before the pandemic. And now we do a lot of Zoom meetings. And we also have wonderful collaborators on campus, outside campus, and around the world, actually. And I'm, of course, very grateful to all the funding agencies who have supported our work over the years. So with that, I thank you so much for your attention. I'll be happy to address any questions you may have.
Thank you, Ling, for the wonderful talk. So now we have some time for questions.
Leo. Yeah. Dr. Li, that was an absolutely fascinating talk. I have made thousands of synthetic peptides over my career with colleagues, and I know the good things and the bad things about them. So the E4 is an interesting molecule. And I have a couple of questions. It seemed to me in the assays you were using, and I may have read them incorrectly, it was functioning at a micromolar level. I think you had it. And, the fascinating thing to me was the fact that it worked as a detergent, essentially, for A beta efflux in the cells, which is kind of exciting mechanistically as far as what one might do with drug discovery. So the question I have for you is that, has that peptide been A, optimized, B, is that a level that could really be approached therapeutically with it, or a more effective analog? And do you know anything about half-life?
Thank you very much, Leo, for your kind comments. And then, I'm so happy to know that we have experts on here, with the peptides. And to answer your question, you are absolutely correct. It's like a detergent, right? But it's all the apolipoprotein, they all have that because the amphipathic helices. Optimization part is indeed in need of micromolar concentration, and it has clearly done in mouse, and it's done in clinical trials in human, and 3 clinical trials already. Clinical trials, it's not always consistent because, the formulation of the peptide. And I want to also mention, there is a trick you can do. I'm sure, Leo, instead of using the L peptide, the amino acid, you actually can synthesize the D amino acid peptide. So with that I would say that's one of the optimization maybe, because the D peptide we have tested, I didn't have time to show, exactly acts like the L peptide in terms of functional wise, but then in vivo, it will resistant to the degradation, so you can have a little bit longer half-life. But that being said, I still want to share with you, this half-life is still quite short, even with the D peptide, because there still can be, filters through the kidney and that non-specific pathway. But I think it is a matter of, like you said, the optimization and in the future, the micromolar concentration, or maybe, we're even thinking working with some folks who are more specific, like more expertise in peptide. So then we might be tweaking that to make it even better.
Very nice answer. Thank you. Again, very fascinating book.
Thank you very much. Let's see, I see the chat.
Yeah.
Okay. All right.
Okay, Ling, I actually have a question. So you mentioned about statin, how that's affecting the prenylation pathways, right? So my question for you is that, do you know how it's actually the statin medication use of it affecting the FT enzymes in the brain?
Yeah, very good question. So most of the statins have two kinds of statins. They're lipid, lipophilic and then not hydrophilic, right? So the simvastatin and these are lipophilic statins. They can cross blood-brain barrier, but still it's not much. I think later actually give the credit to Gibson Wood. He unfortunately passed away a couple of years back, and he actually measured the statin concentration given peripherally and then in the brain. And then the other people, I believe there was a paper from Emory University, even though the level is not so high, but they still could affect the prenylation in the brain. They use the rats, but I would think it's similar to the mice. So that is a good question. Exactly to which extent it might be, might be debatable. But clearly, some statin can cross blood-brain barrier, and they are reports, right? So it's ironically, I guess, well, not ironically, to me, maybe people will argue that, there are reports about people taking statins may have adverse effect on the cognitive function. But I would say those are case reports, very few. If you think about how many people are taking statins, they are huge clinical trials have again and again approved. There are no negative effects of statins on cognitive function. My understanding is that I believe because statins affect both GGT and the FT pathway, so that might be some of the discrepancy. So that's why we are focusing on the FT, because as you can see, the GGT pathway can lead to kind of a negative effect.
Yeah, I have a question. There's a lot of interest in tau protein and the phospho-tau protein in Alzheimer's disease. I was wondering if something like prenylation is involved in the regulation of that protein?
Thank you so much for that question, because I don't have time to talk about it in the talk. I was focusing on amyloid. Indeed, there are, Ken Kozik's lab in California, Santa Barbara, he published a paper a couple of years back in Science Translational Medicine and showed that, inhibition, the FT, they used actually the pharmacological inhibitor in the tau model. And of course, this is not the Alzheimer's model. It's all like a FTD model, because it's a primary tauopathy. But they showed that there's a reduction, clear significant reduction of tau pathway through autophagy and other signaling pathways. So this is my next stage. And then I'm in collaboration with Dr. Karen Ash lab. They have a lot more expertise in the tau pathology. So we would like to continue also looking into, dissecting out instead of pharmacological paths, pharmacological approaches sometimes has other effects, but genetically we can, precisely target what way we would like to look. Thank you very much for that question.
Yeah, thank you.
Well, thank you, Ling. And I think this is has been a really wonderful talk. And thanks for sharing your research with us.
You're very welcome. And then thank you all for I would say that if any of you have additional question or you thought of afterwards, and then you know where I am, I'm right in front of this MTRF, right? My laboratory. So you can find me here or you email me anytime. I'll be very happy to address any questions or collaborations. We always open our doors. Thank you very much.